What I'll be talking about is basically just going ahead and working in very highly scattered light imaging situations. The motivation for this is that the deep tissue optical techniques are developed basically to complement microscopy methods, positron emission tomography, MR techniques, and address situations where those methods are not available. So one example would be I'll talk about is in imaging whole live mice, and this method, for example, fluorescence molecular tomography, provides molecular imaging with all of the advantages of optical contrast agents. In humans, I'll also talk, so another situation I'll talk about is in humans where diffuse optical tomography methods and near-infrared spectroscopy provide a portable bedside technique which is not found with either MR or PET. So here's an outline. I'll quickly go through some of the basics of tissue optics, instrumentation basics, and image reconstruction. And then I'll highlight a few recent mouse imaging systems and a human neuroimaging system. So first, tissue optics. And I'll start with just a little bit of a perspective. As was mentioned in the previous talk, optical microscopy gives you high resolution up to a depth of a millimeter, maybe it goes a little higher, and then suddenly there's this big jump and you lose resolution by almost 3 orders of magnitude when you get to diffuse or highly scattered medium. And so the question is why, and the answer was given by Jorge, which is that this really scattering that sort of turns on here and creates this problem. So first, let's just review what the properties of tissue. So first is what is absorbing. If you look up in the high wavelength or high energy region, or short wavelength region, I should say, you have electronic interactions with tissue. So water has very high absorption, and there's kind of this window. It's clear for our visible region. And then proteins, melanin, hemoglobin. And as you can see, the absorption coefficients are sort of steadily dropping as you move towards this near-infrared region. Then on the other side, you have vibrational interactions with molecules, mostly with water. But in this region in the middle, there's this sort of low absorption window of opportunity, if you will, where you can do biological imaging or deep tissue imaging. The other thing to consider is scattering, and Jorge showed a really nice example of that, so I won't show you, talk about this demo. But I just want to point out that compared to the absorption spectra, the scattering spectra doesn't have this window of opportunity. It's relatively flat, whereas absorption is changing by 6 orders of magnitude or so, scattering is only changing by about 1, and there's no sort of dip in the absorption spectrum, or scattering spectrum. So we have to deal with that. And the other thing to note is that in this, the absorption length, if you will, in this region is almost 10 centimeters. So that's long. You could easily see through your hand if it weren't for scattering. Whereas the scattering constant is at a millimeter. And that's sort of where, if you think back on that contextual slide, that was where we had this quick drop-off or quick change in resolution, was when we went to depths beyond a millimeter. The other property that we'll want to think about is fluorescence. So this is if you shine light in and interact with a molecule, then it does non-radiative decay to a second excited state, and then fluorescence light comes out, and it's always at a lower energy, so it'll always be redshifted. And this is a very useful technique because you can basically do very low background types of measurements where you're filtering out the excitation light and looking only at the fluorescent light. And that allows you to have very high specificity to the molecules or the fluorophores, the molecular reporters that you want to look at. Just to give you a little background on fluorescence, there is some endogenous fluorescence in tissue. It starts with the structural proteins like collagen and elastin, then the energy coenzymes of cellular energy metabolism like FAD and NADH, some amino acids on other proteins, and also porphyrins, which is a precursor to heme. So these sort of span this whole region. One thing I'd like to note is that as you get beyond 700, then the endogenous fluorescence goes to a very low value, and that's another reason to work in the near-infrared region. So in terms of exogenous agents, this is really kind of the main motivation for working with these deep tissue imaging for mice, at least. And this is just to point out that these optical agents are very comprehensive. So this is six different aspects of cancer, and there's optical reporters for all of them. But there's also reporters for many other diseases as well. Okay, so light propagation in tissue. Let's see if this movie works. No. But you can still get the main idea. So the idea is that when you shine light into tissue, it scatters everywhere, and you get this really diffuse pattern. And in a way, that's, the first news is bad news because it's just scattering the light so much. The second news, though, is that this pattern is relatively boring. It's kind of, and that means it's simple, and that means it's relatively easy to model. So we can model light diffusion through tissues as basically like you might model heat diffusion through any other material. And this is a fairly simple equation. It's a second-order differential equation where the first term is a second-order spatial derivative, and that's related to scattering. We have just an absorption term, which is just a product of the absorption coefficient times the light photon fluence, a first-order derivative in time, and then we have the source of light where you're injecting light into the tissue. So the only physical parameters of the tissue that are coming into this equation are just the absorption coefficient and the reduced scattering coefficient. So that's we can simplify this differential equation further if we assume that the medium is homogeneous, just to get started. Then it looks like this. For a time domain, it simplifies even further if we have a light source that has that's frequency modulated, or has a frequency term, or sorry, frequency domain light source. So then we just have basically a second-order spatial derivative and this sort of an attenuation coefficient. This is just the solutions to these homogeneous differential equations. The time domain solution is still a little bit complicated if you look at it mathematically, but if you look at it in practice, it's not that. Conceptually, it's easy to understand that you just have this sort of injection of light and then it sort of falls off depending on how far you've gone through the tissue. The frequency domain solution is actually starting to get simple enough to think about. This is essentially just an exponential decay with a one geometrical term, which is just divided by r. So this is basically just an spherically expanding wave of light, and it's decaying in its intensity the farther away you go from the light source. So what we'll want to do now is use these sort of simple models of how light propagates through tissues to understand how we could put together an imaging system. So the first thing to do is to consider light illumination in tissue. So this is, again, very simple. You just have light injected into the tissue. You're going to get this spherical expanded wave. And then, so that would be essentially how much light gets to any particular voxel. Then there'll be interaction of the light with the voxel. In this case, I'm just using absorption of light in the voxel. And then we need to propagate the light from the voxel to the detector. And so that's what this pattern would be, which is just how much light gets propagated from this voxel to this detector. And so for this, we're actually going to run the problem backward. It's a little bit easier to think more of the sensitivity of the detector to any one particular voxel. And I can use essentially the same propagation function that I used for the source to the voxel to get from the detector to the voxel. So then when I want to consider a measurement, any optical measurement always includes both illumination and detection. I just end up multiplying these two functions times each other, and I get this sort of crescent shape, or depending on how it is, it'll be this it's a sampling between the source and the detector that is sort of wide and blurry, which is why we can't get crisp images like you can with CT, but we can still model them quite simply and put together both mathematical models and just understand how all this works if you want to design an instrument. Okay, so there are other models for light propagation through tissue than that. The simple one that I presented, what I presented on the last couple of slides was a diffuse wave approximation with an analytic solution. You can also do it through more complex geometries using numerical solvers. And then you can go to more sophisticated models that include a little bit of the memory of where the light was going, radiative transport equation. And again, you can use these, solve these with numerical models. As you go to more sophisticated models, you can handle more sophisticated and complex tissue situations all the way up to clear voids such as cerebral spinal fluid or something like that. But that comes at increasing computational complexity in terms of the code and also in terms of the compute times. Okay, so let's move on to image formation. So any diffuse optical tomography system can be kind of to think about how a tomography system is working, it's useful to think about three different aspects. First, what is the data type? Is it just a continuous wave system, or does it have time-resolved measurements, or fluorescence, or you can even use coherence, what I won't talk about too much today. And then there's also what sophistication you have in terms of the reconstruction. You can do reflection transmission measurements, which is kind of what Jorge was talking about. You know, what you see is what you get. Or you can use these indirect models that have either photon migration modeling in them all the way up to 3D tomography. So these are kind of the style, I guess, of the instrument, but in addition to that, you also just have the raw data size aspects of what is your spectral sampling, spatial sampling, and temporal sampling. Okay, so just a few comments on the types of data. As I said, you can have time domain, frequency domain, or you could have just a CW light source like this laser pointer. Again, if you work with the time domain, you have the sort of most complete information about the tissue, but it's the most complex and expensive. And so there's also a trade-off here in terms of, you know, faster, easier, cheaper data, or do you want complex, more expensive data? Then there are different, one of the next questions to answer in designing an instrument is what type of spatial sampling that you want to use. And this dramatically affects your ability to create images. So you can use simple geometry, again, just shine a light on a mouse and take a picture, or you can move to slightly more flexible scenario where you've got a light source, which would be this laser diode, and a detector, a PMT here. And they're both coupled to a pair of fibers up here, and then you relay the pair of fibers down onto a mouse. So this would be raster scanning of a mouse. Or you can sort of combine these two approaches and have a camera viewing the entire surface of a mouse while you laser scan from the bottom of the mouse. And again, as you go this way, they become a little bit more complex, but you gain more information. So I'm going to go back and run through these in terms of the modeling each scenario. Actually, the simplest one to think about is the second one, which is the raster scanning. In that case, we simply are shining light into the tissue at one point, detecting at another position, and we're sampling the tissue with this type of sampling function. In the reflectance imaging situation, we're again shining light in at one point, sorry, let me try this again. We're actually illuminating at many points on the tissue. So we'd have to have this sort of generate a planar illumination source from our point sources, sum over all of these different point sources. And then we're detecting just at one point, which is where the CCD is image relaying a little spot on the tissue to a pixel. Finally, with the tomography, what we're doing is shining light in from one direction, and so it illuminates the pattern like this. And then on the other side of the tissue, we have a detector that's sampling the tissue with a sampling function, something like this. For any particular measurement, we have a source detector pair which samples the tissue like this. And then we have a bunch of these different measurements that sample like that. Or you can look at a fixed detector and all the different sources, and it samples like that. And so if you consider this source detector pair and that source detector pair, you can sort of see how they would triangulate out a deep signal from the tissue. So all of these measurements are then put into reconstruction algorithms to assemble them as you might assemble a CT image. So to do this, I want to run through a little bit how we put together the forward model for an instrument. So I've shown you a bunch of these little source detector pair measurements, but now let's just put them into a mathematical formulation so that we can reconstruct it, or at least model our measurements. So the first thing is the measurements. Most often you work with ratioed measurements for diffuse optical tomography. This might be the light intensity during brain activation versus rest, or this might be the fluorescence intensity divided by the excitation intensity. So that's Y, is just your list of measurements. On X is also relatively simple. It's the 3D tissue structure you're trying to image. In this case, I'm just considering differential concentration of a fluorophore throughout a tissue volume. This is listed as a one-dimensional vector, but really it's a three-dimensional space that we've sort of unwrapped into a one-dimensional vector. And then finally, the propagation model, this matrix, which is going to link the tissue structure with the measurements. So here, this looks kind of complicated, but it's actually relatively simple conceptually. Basically, again, we just have these Green's functions or propagation functions, which take the light from the source to a voxel, and then they take the light from the voxel to the detector. And then we're just, let me go back one last, and then we just are normalizing by how much light goes from the source directly to the detector. Okay, so this kind of wraps up the forward model. We now have a way of predicting what our measurements will be for a hypothetical tissue structure. The next thing we need to do is the inverse problem, as Jorge was talking about. This is an indirect problem, and so we need to, we're going to need to invert or somehow invert this equation. So first, I just want to sort of conceptually sort of simplify what this A matrix is doing. It's basically making a map from image space to measurement space. In a normal system, this is kind of hard to visualize or conceptualize. So, but you can do it if you really simplify the problem. So here's a very simplified diffuse optical tomography setup where you have a source, which is the one light source and a whole row of detectors, and then we're only going to try and image just in this one row of voxels. In that case, that row of voxels goes to every column in this matrix, and then this row of detectors goes to every detector or every row in that matrix. So this is kind of a simple mapping of from this voxel to all of these detectors, and that's what this image space to measurement space matrix would do. So now when we want to do inversion, we basically just want to, change that equation from y equals ax to x equals a inverted something times y. So we strictly if we only cared about computation, we could do that. We can invert the matrix, but it won't work because the measurement noise is going to corrupt the image. Basically, there are very tiny elements in A due to this diffusion problem that when you invert the A matrix, they become extremely large and they'll corrupt the images. So this has to be managed somehow, and the way you manage it is through a process called regularization. So I'm not going to go into detail about how regularization works, but basically we have to manipulate the inversion of this matrix so that we can control noise in the image, and when we do that, that introduces a smoothing operation. And that's where we end up, sort of mathematically, that's where we end up losing resolution. So in the end, we always end up with these regularization parameters that control the amount of regularization, the degree to which you're controlling image noise. And this regularization parameter will control the relationship between image noise and resolution. And so this is just a sort of a recap of all of that. Again, we have a forward model, which is just basically a matrix which will map from tissue structure to measurements. And then we have to invert the problem. And another way, kind of conceptually, to think about this inversion problem is that it's a fitting procedure. So we are going to fit, and we are going to do that by minimizing the error between our measurements and some model that we have. And the parameters in the fit are all the voxel values. So this is the error function that we would normally minimize in just a simple fitting procedure. And the regularization comes in terms of penalizing the amount of noise that you would have in the image. And so, you know, in a way, inversion is there are many tricky things to inversion, but at its sort of bare essence, it's really not that complicated. Okay, so now I'll just move on and show you some different imaging systems. I'll start off with these, a couple systems that are used for mouse imaging. So I showed this one actually, the reason I'm showing it first is that it kind of mimics the mathematical description I've had of the tomography systems. Basically, a laser source is coming in, it goes to a galvo pair of mirrors, and then the laser is just shined or is shown onto this surface here, and then it can just be raster scanned to fill whatever pattern that you want. And then on the other side of this tank, we're imaging the light that's being emitted from the tank using a camera. And so this is sort of a close-up from the source side of a mouse being put into this tank. And then using, in this case, we were using intralipid as a sort of a matching fluid, if you will, so that the entire region of the entire area of the imaging domain was all diffusive. So the mirror scans with small spot size, much smaller spot size than the resolution of the system. And let's see, and then the camera is just low noise and has you want these to have high frame rate because then if you've got to scan for all these different source positions. And so when you put all this together, you can do some simple phantom experiments like image a couple tubes, and it maintains resolution, and sensitivity is relatively flat across the whole imaging domain at about, for this tank, at about 2 millimeters for a 15 millimeter sort of distance. And you can get nice linear response or linear quantification of fluorophores over 3 orders of magnitude. And you can calibrate them so that you know exactly how much fluorophore you have in any particular place in a mouse. Here's an example of just using it to image a molecular imaging agent. This is just in a simple model of a subcutaneous tumor on a mouse, and this fluorophore was targeting mammoglobin receptors. Here are a couple other systems similar in that they're both planar tomosynthesis systems. This is one of the earliest systems put together by Vasilis, and it's pretty similar to what I showed on the previous slide, except for in this case, the sources, instead of being scanned over the source side of the imaging domain, it just uses a bunch of fibers which are then switched between to sort of run through all the different source positions. Here's a more recent example. Again, light comes in from one side. You detect light emitting from the tank or cassette on the other side. And the nice thing about this system is it's got this really nicely designed cassette so that you can put a mouse in the cassette, image it with FMT, and then take it over to a CT scan and get CT data. So this is a way of starting to add CT data into FMT reconstructions or FMT studies. This is one of the most recent examples of FMT. And in this case, it's been completely integrated with whole body X-ray CT. And the imaging for the FMT is somewhat similar in that source comes from one side and you image on the other side with a camera. But now they're going to spin, in this case, this source and detector are spun an entire 360-degree revolution around the mouse, so you have a really nice data set to work with. It's all co-registered with CT, and so the first thing you get is just that the fluorescence molecular images are co-registered with anatomy, and that provides a reference for understanding where things are accumulating. This is FMT images in this column, and B are the same mouse, but this is cryohistology. So this is the ex vivo kind of validation that these fluorescence contrasts are really where they are supposed to be. This is just showing another example of the power of having the X-ray data co-registered with the FMT. You can segment the CT data to give you the bone anatomy of the animal, and that gives you a better idea, again, of where the fluorescence is coming from. And it goes further. And not only is the FMT data co-registered with the X-ray data, you can actually combine the X-ray anatomy and use it as a prior information to improve the FMT reconstruction, and that's shown here. And one of the things that you can, there's a couple different things that you can get back from that. One of them is that you can improve the resolution. So this blue curve showing the FMT reconstruction with just FMT data alone. The yellow curve is showing the improvement in resolution if you include the X-ray data. Okay, so this is another system, again, combining FMT data with X-ray and then also with SPECT. So this is basically a fiber, this FMT system is now a fiber-based FMT system. It's got blues are sort of like for the sources, the reds are for the detectors. And this, by being a fiber array system, it now works as an insert. You can put it into any sort of commercially available SPECT, CT, or PET type of system. So here's the CT data. Here are the fibers that show up in the X-ray data. You can use, again, the X-ray data to create an anatomical model with which to do the FMT reconstruction. And then you get both FMT data in red and SPECT data in green. This was a injection into a forepaw and the drainage into the sentinel lymph nodes. And then the yellow shows the overlap between the two different imaging modalities. The other advantage of this fiber array system is it uses discrete detectors that are very fast, and so the whole imaging time this whole system runs at 30 hertz. And so it's fast enough that you can image injections of contrast agent. So this is just an injection of indocyanine green. It peaks at about 10 to 20 seconds, and then because we can image that fast, we can image the accumulation of this contrast agent in the heart and use the heart as basically an image-derived input function for doing kinetic modeling within the mouse. So this red curve is the our arterial blood sampling that would be kind of the gold standard that we'll judge ourselves against, and this is blue is how well we fit against the gold standard. So now that we have this image-derived input function, we can look at different regions, for instance, the lungs or the liver, and try and derive the kinetics, quantify the kinetics between the blood compartment and then these different organs. Okay, so the last thing I want to talk to you about is imaging in humans. And here the idea is to do imaging like functional connectivity or like functional MR, but do it in places like the operating room where it'd be difficult to bring in an MR machine. So the way it works, again, is you have source of light injects into the head, you have a detector, and it detects light somewhere away, and you get sampling that reaches down to the cortex. The improvements that have occurred in the last 6 years are that the instrumentation has been developed to get sort of these high-density arrays, high density of sources and detectors onto the head, and also some improvements in anatomical modeling that is similar to what we saw with the mouse. The challenges for optical imaging of the brain is, again, lateral resolution. Resolution is always a challenge with diffuse light systems. Also, there's challenges with removing the systemic artifacts that occur for sort of the dynamics of blood in the scalp tissue and skull from the brain signal, so getting sort of a brain-specific signal. And then finally, the other challenge is just creating a system that has a large enough field of view so that you can cover a decent amount of the cortex. So sort of the main advance that we've been working on is just moving some really simple ideas just to move from sparse arrays to high-density arrays. So the green lines are measurements. More measurements are good. And basically the lateral resolution that we gain in lateral resolution is really just due to having a more dense sampling of green lines. So it's easy to just make this PowerPoint figure instead of that PowerPoint figure. The hard thing that all of the groups had to do was to deal with the dynamic range that is asked for when if you draw all those lines, then you have to have a dynamic range that's like this, which spans 6 orders of magnitude, and this whole thing has to run at a hertz because the brain dynamics are quick. But a number of groups have started to build these types of systems. These are essentially all of the high-density neuroimaging papers between 2000 and 2010. So groups are starting to bring them online, but it's still a pretty new field because there's only about a paper a year. So that's the instrument. The algorithms are starting to look like this. We bring in different types of MR scans, T1 and T2 scans, so that we can segment the head volume into different tissue types. And that gives us a mesh to do anatomical reconstructions, and where we bring in our optodes. So not too different from what's going on with the mice as well. It worked. So if you put together the instrumentation with the anatomical algorithms, and you can get the imaging to work like this. So so first the study is a subject just sits and looks at a computer screen, and this flickering checkerboard sort of rotates. And as it rotates, it's mapping out the visual field of view for that subject. The activation is back in the visual cortex, and this movie is sped up by about a factor of 10, so this really takes about 30 seconds to go around. So this is the response in the increase in oxyhemoglobin on the back of the brain, and this is the fMRI bold response in the same subject imaged on a different day. So this is kind of just to go back to this. So with small animals, you can validate by doing things like cryoslicing, which means, you know, when the mouse is done, you just kill it and slice it up. So for human neuroimaging, you don't really have that type of situation. You have to find another way to validate. So one of the nice things about doing functional responses in humans is that it gives you a way of sending test patterns into the brain and looking at if you get them back the way that you'd think you would get. And the visual cortex is a particularly nice place to do that because you can put different patterns on your computer screens and then get different patterns in the brain to test things like resolution, accuracy, and that type of thing. But if optical is really going to be a surrogate for MRI, it needs to have higher coverage. Particularly, it needs to have enough coverage so that we can sense higher order distributed brain functions. So the most recent system that we've been working with has this type of source detector arrangement, and it can cover not only the visual cortex, the motor cortex, and auditory cortex, these sort of sensory areas, but it also can cover some of these cognitive areas as well. So in terms of looking for higher order distributed brain function, we started looking at language processing. So we can have, there's sort of a hierarchy to the way the brain processes language, and we can map it out with different types of tasks. So we can have people hear words, we can have them do covert reading of words, we can have them imagine speaking words. So here they're just looking at a word, they're saying it into themselves internally, so they're not speaking out loud. And finally, we can have them do covert verb generation. So in this case, they see a noun like dog, and they have to come up with a verb that goes with it, like kick or bark, or, you know, depends on whether they're nice people or not. And then this activates this, since this is really just asking the brain to process from one type of word to another type of word. This is a true language task, and this activates Broca's region. So these are the images that we have averaged over 8 subjects. We take the same 8 subjects and scan them with MR, and we get these maps. So this allows us to sort of, as in the mice, we would be co-registering fluorescence markers against histology here, or fluorescence markers against PET. Here we can judge oxyhemoglobin maps with optical against fMRI maps with bold. Turns out, of course, some people may be familiar with this, but it turns out, of course, that you don't have to ask the brain to do things for it to be doing something. It's always doing something. And so you can map the brain by just looking at its resting state fluctuations by looking at correlated structures in those time series movies. And so here are some results from that. If we put a seed in the visual cortex, it maps out the visual, the other contralateral visual cortex as well. If we put a seed in the left auditory cortex, it picks out the right auditory cortex. And if we put seeds in these other more complex, high-order networks, such as the dorsal attention network, or what's the default mode network. There we go. So the default mode network is a network that is composed of self-directed, internally directed thought, so it's sort of the daydreaming network. When we put a seed up here, you get these constellation of 3 seeds on both sides of the head. These networks are a little bit more advanced, or a little bit trickier to pick up than these, because they have not only bilateral connectivity, but also anterior-posterior connectivity. And these are the same subjects again with fMRI. Okay, so I'll conclude just a quick recap. The forward problem in terms of doing diffuse optical tomography, the whole goal is to get this sensitivity matrix. And the methods that include using the diffuse wave approximation or numerical solutions to that, or you can do more complicated transport models. For brain function, typically the simplest analytic semi-infinite diffusion solution has been used, but more recently we've been using anatomical methods up there, or anatomical information with numerical solvers. The inverse problem, the goal is to invert the A matrix, and the methods include direct inversion or iterative technique, and the main thing to remember is that regularization optimizes the image noise versus resolution. And finally, just to recap sort of the motivation for the optical methods, methods are being developed in the context of things like optical microscopy, PET, MR, and they're really being developed for places where those other methods do not work that well, such as in the mouse, where we have molecular imaging in live animals, and in the humans, where we can do things in situations like the bedside, which would be hard to do with PET or MR. And with that, I'll conclude.